In this lesson, we're looking at passive and active immunity. So specifically, we've got a compare here, remembering that compare means find similarities and differences, and we're also interpreting some long-term response data. All right, so immunity can be classified as produced naturally or artificially. So it's not a judgment of its quality at all, and regardless of whether it is naturally or artificially acquired, it can be passive or active, just like our learning. Now, our focus specifically is on antibodies produced to specific pathogens. So if this is our dot point here, we're looking at active versus passive, and both naturally and artificially required. And so we're going to classify them in this way with these diagrams here. Now, passive immunity basically means we're accepting, or passive in general means accepting or allowing what happens, what others do without any kind of active response or resistance. So the antibodies that are used in this system are acquired from somewhere else without the body having to actively produce them themselves, okay? So antibodies are made in different organisms and they can be transferred into us. Our body's not doing any heavy lifting. So generally they last a lot less time than if we were producing them ourselves, and they can degrade in the system, in our blood system, and they can't be replaced. So very first one, passive immunity, we're just talking passive. This is naturally acquired. So in terms of antibodies, they can pass from the mother to the fetus via the placenta during pregnancy, and then once the baby is born, they can receive it via breastfeeding. Okay, until an infant's immune system is able to actively make these antibodies, they're really, really vital. And we can see that we've got time here and we've got the fraction of adult level of serum immunoglobulins. So obviously there's going to be before birth, and then eventually baby will produce their own antibodies. So if you follow the antibody levels, you can see the passive transfer is quite high, and then it's going to take some time, months to years, before an infant can actually make a comprehensive variety of antibodies. Okay, another type of passive immunity here is artificial. So while the body's still receiving antibodies, which is, made in another source and transferred over, this time they can be made purposely. And then preformed antibodies are specifically made to a certain antigen, and they can be made in a lab and then actually injected into someone. So an example of this is snake antivenom. It's used to treat snake bites, which makes total sense. But in terms of COVID-19, initially when people were quite sick, they were using plasma transfers to actually give someone who is sick antibodies from someone who has made them. So there are quite a number of different ways that this is used, but it's not used often in most people in most cases. Right, active. Obviously, the opposite of passive, the body actually has to make them themselves. We're talking antibodies still, and this can be done through training, through exposure to antigens, and memory B and memory T cells contribute to this response, as you would expect. So active immunity, our body's making those antibodies, naturally acquired. They're produced using the adaptive immune response, right? Using the humoral response with B cells. B cells are the ones producing the antibodies. So it's through that humoral immune response, and antigens or pathogens have got to pass through that first line of defense in order to trigger the B cells to produce these antigens. We know this. These antibodies are specialized. This response is trainable, so the body will see that first exposure and then make more and more antibodies based on more types of antigens that it sees. Active immunity, but artificial, is due to the body being purposefully exposed to these antigens in order to train it to produce certain antibodies. And vaccinations do this, right? It's also known as immunization, but this is the express purpose of a vaccination, is to show or expose your body to a certain pathogen to make antibodies. Now, vaccines contain an altered or a weakened or a killed pathogen, so it might be bacteria or a virus, it might be inactivated toxins or a protein that the pathogen would produce, and they're really highly specific to the antigen that the body would encounter if this was exposed in a natural situation. So that antigen aspect has still got to be present so that our body can make its own, actively, antibodies. And vaccines challenge the body to produce both antibodies and memory cells for future exposures. So it's not just the initial antibodies, it's about getting those memory cells as well. Otherwise, that trainable aspect doesn't exist. So multiple vaccinations may be required in order to train the body to produce enough of a strong response. Okay, so for example, I seem to keep losing my immunity to measles, so I've had to have multiple vaccinations so that my body keeps retraining itself. And so if we look at this vaccination schedule, you will see a lot of double ups. So we see a lot of different, hey, you need to get this 3 or 4 times in order for your body to be properly trained. So babies don't start getting vaccinations until they're at least 6 weeks, mainly because maternal antibodies can still be circulating, and therefore, you know, anything that might produce, you might actually have some handy passive antibodies that can work to kick in an immune response. Now, immunisation programs are obviously in place all over the world. Some vaccinations are effective with that single dose, others are provided in multiple doses, or you have a booster after 10 years or something like that to ensure that those memory cells continue to circulate. Pathogens which evolve really quickly, like the flu, mean that we need to have a new vaccine made every single year to keep up with the changes. So flu vaccines are usually a mix of antigens of various flu strains that have been seen in the last 12 months, which is why getting it too early might not be helpful. Getting it a bit later, you might see some new antigens put in the mix. But, you know, getting the flu shot doesn't necessarily mean it's going to protect you for the next one, but it's going to hopefully prepare your immune system so it can put up a fight. Now, herd immunity is really, really important here, and it involves a sufficient number of people in the community being immunised against a particular pathogen. And this is going to allow people who are unable to become vaccinated, like infants, like older people, like immunocompromised people, pregnant people, whoever, to be protected, knowing that the people who have become exposed to that pathogen will actually be able to mount an immune response to it, rather than just spreading it on willy-nilly. So booster vaccines, might not be taken, people forget, they lose track of when they last had one, and it can lead to this decreased herd immunity in a community, all right? And so can vaccine removal. So things like whooping cough, it's caused by a bacteria called Bordetella pertussis. It's vaccinated against as a baby, but people often don't get the booster, and therefore incidences of this are increasing considerably. Same with measles, which is something we don't usually see in communities and is now starting to pop back up again. So when we look at long-term response data, it shows us that vaccinating as the first exposure means that the second exposure results in a larger antibody response, and it can occur when that pathogen actually naturally comes into the body. If you don't have that first vaccination exposure, then your body at the first exposure is probably going to suffer because you cannot mount that huge immune response first time around. If we focus on comparing active and passive immunity, please note there are far more options than what I'm providing you here. Similarities, both of them can occur naturally, both of them can be artificial. We can see that in what we've talked about already. In terms of differences, antibodies are created in active or just received in passive. That's the definition of active versus passive, right? The antigens are trained in the system, in the active. There's no training happening. It's not a trainable response in passive. You're just receiving them from a different source. Memory cells are produced when you actively produce your own antibodies. There are no memory cells produced if you are simply receiving passive antibodies. And there's a long-term response and a short-lived response. So if you are producing memory cells, you would expect that there would be a long-term response, but only going to use the antibodies as you have them, and then they will degrade in the system in terms of receiving them passively. So again, comparing active and passive is the main point here, and you really do need to understand your specific adaptive immune response to understand what's going on with the active part and understand really what antibodies are in the first place.